Thank you, Professor Chibale, for the insightful and exciting talk. And we'll go ahead with a Q&A session. Hope you're okay to go ahead with the questions. Please confirm.
Good.
Yeah,
Yep,
thank
I'm
you.
good. Thank you.
Yeah. So as there are a lot of questions, but we'll try to take as much as possible from the audience. So to begin with the first question that we have you screened larger medicinal chemistry libraries in the kinase-based assay?
Yeah, so thank you for that question. So we focused on screening very, very targeted libraries. So the ones that we've screened so far, apart from the 84 compounds that GSK screened, we've also screened a library called the kinase chemogenomics set. So this particular library it's less than 200 compounds, but it's very highly annotated. And so we have a lot of information about the human targets. And so we've screened that library, identified chemistry starting points, and we've used that to also then do mode of action deconvolution to identify novel kinase targets. So the answer is yes, but very focused screening.
Yeah. So thank you, Professor Chibale. And the question again related to the fact that the kinases that you have tried to target, they are against the Plasmodium falciparum, not against the human kinases. So please confirm that was taken in your database.
Yeah, so the two kinases that we found that are implicated in, for example, the story I gave you of sapaticity, those have human orthologs. And that's exactly why we are using, thankfully, there are a number of protein structures of human targets available. And we're exploiting the differences in the structure of the human and parasite enzymes. For example, in the targeted covalent inhibitor approach, for example, we find that in the PI4K, the phosphoinositide 4 kinase, there is a cysteine in close proximity to the ATP binding site that is not in the human ortholog. So we can exploit that difference to then design inhibitors that are selective for the kinase or from the parasite over the human one here. So yeah, so that's what we're doing. Really, it's really the ones that we've identified also have human orthologs.
Thank you, Professor Chibale, for clearly highlighting the distinction between both the kinases. And we'll move to the next question. You talked about one of the anti-cancer component or compound, we can say, spanisertib. And the question is around, have you studied the parasite clearance time, or let's say, how much time does it take, or on the kinetics basis of a spanisertib in comparison to that of artemisinin, the gold standard that we have for malaria? Yeah.
That's a great question. So first of all, one of the things that we use to rank, if we have many chemical series and we want to prioritize which one we should we should work with first, all things being equal, we look at the killing kinetics. So we look at the killing kinetics in vitro. We call it the parasite reduction ratio. This is basically how quickly the parasites are killed. And then, of course, something else we do when we're doing in vivo studies, we can also see how quickly the compound is working in vivo to clear the parasites. And then, of course, when you talk about the artemisinins, you see that in the clinic, they are very fast killers. But the example I'll give you is many of the PI4K inhibitors, the compound that I referred to earlier. And this is a very big lesson. When you look for the killing kinetics in vitro, it's actually quite slow. In vivo, it is just moderate. But when we went to phase 2 human trials, they were as quick as the artemisinins. So, there can be a disconnect between what you see in vitro or even in a mouse model compared to what you see in real people. So the answer is yes. As one of the progression criteria, we always do killing kinetics and obviously look for things that can kill the parasites quickly, but unlike the artemisinins, also have a long half-life because the artemisinins have a very short half-life.
Yeah, thank you, Professor Chibale. So again, coming to the targetability and druggability of the kinases that you have talked about. So you have mentioned that Plasmodium kinases are great drug targets for malaria, and which is great for the treatment of infectious disease. But what are some of the challenges of Plasmodium kinases as targets? So on druggability, you did highlight some of the aspect, but when we talk about the targetability, how do you take it?
Yeah, so I think the one challenge with malaria kinases, and this is true about many malaria proteins, is that they're very difficult to crystallize. They're very difficult to work with. For example, to this day, and we've tried with our partners, with Novartis and other people, there isn't a crystal structure of the PI4K, that lipid kinase. We still, everything we've done with the structure-based design is using the homology model. So they are very difficult to work with, very difficult to crystallize and purify. So when we do cloning, expression, and purification, we get very low yields and they're very badly behaved. So that is one of the major challenges with malaria parasite kinases, is that then it doesn't really help us with when we want to determine the co-crystal structures, X-ray structures of the protein in complex with the inhibitor. PKG, the other one I showed you, that has been
Yeah,
successful. We've got many structures, but lipid kinases, very challenging.
yeah. So So, Professor Chibale, thank you for covering that part around that. It's very difficult to get the crystal structures, and that was one of the questions. So we'll move to the next part, and it talks about what is the activity of these molecules against resistant cell lines. So the molecules that you talked about, let's say, in the differentiation around the kinases and hematinib. So
Yeah, so
yeah,
they are basically equipotent. In other words, um, I use the term cross resistance. So it means that if you look at chloroquine, in a chloroquine sensitive strain and in a chloroquine resistant strain, if you have a compound which shows the same pattern, so chloroquine is more active in the sensitive strain and less active in the resistant strain. So if you have a compound that has the same profile, not necessarily the same activity, but same profile, then there is cross resistance. But in this case, to answer that question, they were equipotent. So the data I showed you on DD2, which is a resistant strain, was about 70 nanomolar. In the sensitive strains, it was about plus or minus 4. It was within that range. It could around 60 something to 7. So equally equipotent across both strains. So there was no cross resistance, and that's exactly the point. Is that the compounds were active on resistant strains that are known already.
so Professor Chibale, we do have a few more questions, but I would like to request if I'm allowed to take one more for you. If
I
the
am
time
uh,
permits,
I am
yeah.
available for another 100 questions if they are.
Thank you so much. So we'll take two more quick questions, and uh, the f- the first one that I would like to take is it's a shorter one. Are the Plasmodium kinases suitable for cryo-electron microscopy?
Well, again, this is part of the challenge I was talking about earlier. That they are very. We actually been doing an experiment with a colleague overseas to try to use cryo AM to do that, and I think there's been, it's been successful with. There's this protein called. It's a protein. It's a transporter that's been implicated in chloroquine resistance. It's called the Plasmodium falciparum chloroquine resistant transporter. That is not a kinase, but it was really, the structure was solved by using cryo-EM. To my knowledge, to this day, we have not managed to get a cryo-EM structure of our kinases. They've still been challenging, and this is another challenge. Just the techniques that are available are not really helping us with solving the crystal structures. It's not true for every protein, because you saw the PKG, the protein kinase G, that's been solved. It's only these lipid kinases that are very badly behaved.
So I'll come to the last question, and it looks like a concluding thing. So how do we address the cost of therapy associated with the malaria with kinase inhibitors? And that's one of the things where your take is important. Yeah.
Yeah. So first of all, when we talk about target product profiles, we think about the patient, and one of the important target product profiles is the cost of the medicine. the cost of goods. Now, one advantage of the approach of using polypharmacology, so if you look at malaria treatment or TB treatment or cancer treatment, drugs are given in combination. So you select drugs based on how you know they work, based on different mechanisms or targets, and then you combine them. Now, the cost of developing a combination regimen and combining different things adds to the cost of the medicine. Now, imagine if you are able to find a single molecule that hits multiple targets. That is like having a combination therapy without taking individual molecules that act by different mechanisms. So that is one advantage, yeah. But the other thing that is an advantage potentially is when it comes to polypharmacology, which can be an advantage in addressing selectivity over human orthologs, is that actually you don't need very potent inhibitors. So most kinase inhibitors that bind in the ATP binding site are very potent. Talk about nanomolar potencies. But actually, if you are going after polypharmacology, you are also relying on synergy. So you don't need very potent inhibition of a single target. You are relying on the synergy that arises from hitting more than one target. So that is why this approach of polypharmacology in the context of malaria is also could have a potential advantage of the low cost of goods. But of course, we've only developed one kinase inhibitor that went into the clinic. It didn't progress all the way, but it went to phase 2. But before we could progress it, the cost of goods is important. And that's where the chemistry comes in, yeah? So that compound I showed you is only 3 steps. You make it in 3 steps. 2 Suzuki cross-coupling reactions. And just 2 Suzukis and 1 other reaction. That's it. So it's not just the mode of inhibition, but it's also the number of synthetic steps that are involved in making the molecule and the reagents that you use that all contribute to the cost of goods.
Yeah. So thank you, Professor Chibale, for taking up most of the questions and sharing your expert thoughts. Now, I would request you to give your final remarks before we close the session. So final remarks from you, please. Yeah.
No, I just, all I can say is, we are talking about all of us here are chemists, and I think that to the students who are training in drug discovery, I think it's I was trained as a synthetic organic chemist, and I think the beauty of doing drug discovery is that it really is not just, it shows you lessons in teamwork because it's a team-based discipline where you cannot do it without having the other disciplines together. But it shows you also that in life, there is not a single discipline that more important than the other. All disciplines are equally important. But it's also an avenue which allows you to really learn a lot. I'm still a chemist, but I've learned a lot from biology because it's in the context of a particular program. And I think the other message I think I'd like to give is that we have to understand that we have to fight for chemistry. And what is the challenge for all of us is how can we show that chemistry is important? There are many things that are important. So I think what we have done, even just the few slides I showed you introducing a drug discovery center, when we started, we only had 5 people, 5 postdocs. Now we have 76 people. Now, what is the message here? The message is very simple. This is about scientific entrepreneurship as well, that we can show to our governments, whether it's the government of India, the government of South Africa, wherever the governments are, that the business case for chemistry is that it's not just that we can solve human challenges. We may not even discover a drug in our lifetime. It doesn't happen easily, but we can show to our leaders that chemistry underpins what we do. We can create jobs. We can build infrastructure. So don't underestimate entrepreneurship. And entrepreneurship is not just getting money from a bank and starting a business. It's also creating infrastructure that allows people to practice things like drug discovery, which is underpinned by chemistry and biology. So that is my message, is that let's show that chemistry is important, not just because we can solve problems, but we can create jobs. Yeah, that's a language politicians understand, jobs. And if you can help politicians create jobs, they will help you because they like to cut ribbons and appear on TV, and they can talk about what you did. So that is my message, my parting comments. Thank you very much indeed.